So now that we understand how the process of transcription and translation works, I want you to think about that whole process as being the expression of the information contained in the genome. So you take this encoded information and you use it to make something. In most cases, we're talking about a protein here. Okay, so this is the process of gene expression, taking that information and making whatever it contains the information for. Okay, so in using my recipe analogy, it's taking a brownie recipe and you end up with brownies. That's expression. Now, what we're thinking about now is when does that expression take place and what regulates it? Because as I've told you, you have tens of thousands of genes in your cells, and all living organisms have many, many genes, but they certainly don't express all of them all the time. They certainly don't transcribe and translate every gene constantly and at the same rate. One of the examples I used earlier in this class was the production of alcohol dehydrogenase if I have consumed some alcohol. If I haven't consumed any alcohol, there's no need for me to produce alcohol dehydrogenase, and I will not transcribe that gene. I will not have any messenger RNA to translate. Therefore, I won't end up with any of that protein, simply because I don't need it. And so that's exactly what we're digging into now, is what mechanisms are employed in order to determine when and if genes are transcribed and translated. So, in order to do this, we start out this chapter with some terms that can really be a bit of a struggle for students to wrap their heads around. Okay, so I encourage you to pay close attention to what I say in these next two slides. I encourage you to read this section of the text, 15.1, very closely. I encourage you to look for and access any resources available to you on the internet or YouTube, whatever, in order to help you understand these terms. Okay, so the first three terms that we're going to talk about, inducible, constitutive, and repressible, these terms explain the way in which gene expression is regulated for a particular area of the genome. Okay. Now, if a region of the genome contains a gene, and it will have what we call regulatory elements that allow that gene to be transcribed and translated under certain circumstances, we would call that inducible. So basically what this means is this gene is sitting there waiting and ready, if needed, if called upon, to be transcribed and translated. This is similar to the alcohol dehydrogenase example that I've given you. I don't need it all the time, but if I do need it, I can induce transcription of this gene product, the production of this protein, alcohol dehydrogenase. Okay, so a gene that is classified as being inducible is one whose gene product will be produced if that system is stimulated, okay? And there's usually some specific molecule that initiates or stimulates the transcription of that gene. And most of the time, it's a molecule that's related to the function of the protein that would result from transcription and translation of this gene. So for example, you can imagine how alcohol, when it interacts with certain cellular components, whether they're proteins, enzymes, or other molecules, when the alcohol interacts with those, it can kick off this process whereby the gene for alcohol dehydrogenase is expressed, okay? Because you only need to express it when alcohol is present, so therefore alcohol is a likely inducer, okay? Something that will induce expression of the alcohol dehydrogenase gene. Other genes are classified as constitutive, okay? These are genes that are consistently, always transcribed and translated at a steady pace, a steady rate. You're always needing these particular gene products, and so transcription is happening all the time. You don't need to turn it on, you can't turn it off, it's just making it constantly. Okay? And the third category here is the opposite of inducible. This is a repressible mechanism of control. And under this type of control, a gene is consistently expressed, so transcribed and translated, unless a molecule interacts with that locus and shuts down transcription. Okay? And so in this case, you can see it's usually shut down by the presence of the molecule that the system is producing. So it runs and produces a particular molecule, but if you end up with so much of it around that some of it interacts with the gene region itself, it can turn off the production of it because you've obviously got enough of it in the system. Okay? So these are genes that are in this order as presented here. You can turn them on, they're always on, or you can turn them off. Okay? Now there are two other terms that are applied to genes, and these are negative and positive control. Okay? So this can be applied to either inducible or repressible systems. Okay? But negative control, when a system's under negative control, expression occurs until turned off by a regulator molecule. And we'll dig into this. Like I said, I encourage you to read the text, do some more digging. Positive control is a system that expression happens only if some regulator molecule stimulates RNA production or transcription. Okay. Now this may seem redundant with what I had on the previous slide, but it's not. It's saying something different, but it's quite nuanced. And I find it most effective if students do a bit of digging to ensure that they really understand the differences here. I've got some more slides later on in the presentation that might help make this more clear, but for right now, this is where we are. Okay, now what we're going to start out with is what is a very simple regulatory system in bacteria. And the system that we're considering is one that plays a role in the metabolism of lactose. Okay? Now, this is an inducible system. Again, you're going to have to go back and make sure you understand these terms, but this is a system that can be turned on. Okay? And you'll note here that I indicate that lactose, this top molecule, these two rings held together, that's lactose. And lactose serves as the inducer. It induces transcription of a gene region. Okay? All right, so we have lactose, and bacteria metabolize lactose, breaking it down into its two constituent components, galactose on the left and glucose on the right. So bacteria break down lactose using this molecule in the middle, beta-galactosidase, or beta-gal. So this beta-gal is what is used to break lactose into the two parts because the bacteria can use glucose for their metabolic needs. Okay? They can't use lactose, they can't use galactose. What they need is glucose. If they've got lactose, what they need to do is break it up so that they can have glucose and use it for their metabolism. So this is the system we're going to discuss. This seems very simple, right? You've got a raw material, you make some beta-gal to break it down into the things that you can use. Simple, right? Well, that's not where we're leaving it. We're digging into this much deeper, okay? So here is this inducible system, and we are going to discuss what is called here the lac operon. You see it down at the bottom. This term operon describes a unit of genes and regulatory regions that operate together. Okay, And so this operon consists of a regulatory region and the structural genes. So the structural genes are actually the genes with these three-letter codons, three nucleotide codons, that code for very specific polypeptides that are then folded into functional proteins. Okay? So the regulatory region is broken up in this slide. We're talking about a great deal about the promoter region and the operator region within the regulatory region. And then among the structural genes for the lac operon, there are three structural genes: LAC-Z, LAC-Y, and LAC-A. And these are responsible, when transcribed and translated, for the production of beta-galactosidase, permease, and transacetylase. Okay? Now, the purpose of these three gene products, the proteins that are produced, beta-gal, permease, and transacetylase, those three proteins, I already shown you what beta-gal does. It breaks lactose into galactose and glucose. Permease is a gene that allows that lactose to permeate the cell, to come into the cell so they can break it down and utilize it, right? It's not normally just pouring into the cell. So if there's lactose out there, create beta-gal to break it down, create permease to let it in. Transacetylase is thought to function in basically cleaning up the garbage afterwards, but we won't talk a whole lot about that. Okay, so we've got three genes here that are all involved in the same process, and that's likely the reason that they're organized together into this operational unit or operon. Okay. Now off to the left you see a separate repressor gene region indicated on this DNA molecule, and this is physically separated from the lac operon. It might be right next to it. It could be thousands and thousands of nucleotides away. It doesn't really matter. It plays an important role, but we'll see why its location doesn't really matter. These other five elements, the two regulatory regions and the three structural genes, those all must be physically together, as we will see evidence for here shortly. Okay, so when we talk about the regulation of these genetic regions, we are going to talk a lot about cis-acting sites and trans-acting molecules. So cis-regulatory elements, trans-regulatory elements. Okay? Now, this has to do with regulation of transcription of the structural genes, Z, Y, and A. So when we talk about cis and trans, we're talking about things that affect whether Z, Y, and A are transcribed. Okay? That regulatory region does not get transcribed. The repressor gene doesn't get transcribed. The only thing that gets transcribed are these three structural genes. Okay? Now, a cis-acting site, you will see, is a regulatory region adjacent to the gene whose expression it controls. So the cis-acting site here is this regulatory region, the promoter and the operator. They are right next to the genes, Z, Y, and A, whose expression they play a role in regulating. Okay? Now, a trans-acting molecule, this is something very different. This is a free molecule that's floating around in the cell, and this free molecule will bind to cis-acting sites, so the promoter and or the operator. These trans-acting molecules bind to those sites, and they contribute to the regulation of expression. Okay? Now, that's why I said this repressor gene can be right there. It can be way down the line. It doesn't matter. When it's transcribed and translated, it's not the repressor gene we're interested in. It's the protein, the repressor protein that's produced, that is the trans-acting molecule. It is going to interact with the regulatory region. Okay? So, we have these three structural genes. Again, these are the three that are going to be transcribed and translated. So transcription results in this messenger RNA. Ribosomes slide along it and translate it, as you have learned. These three genes that have related function. They both function in the metabolism of lactose, are produced as polypeptides through translation, but you can see the ribosomes drop off each of the polypeptides as they are completed. So once it gets past the beta-gal gene, it drops off that polypeptide and starts synthesizing another amino acid chain that will become permease, and the same for transacetylase. I've spoken to you a bit about the role of lactose in bacterial metabolism, and that they need to break it down in order to access the glucose that they can use for their metabolic purposes. The structural genes, their function is described here. So LAC-Z is responsible for producing beta-gal that breaks down lactose. LAC-Y codes for permease, which allows the lactose into the cell. And I already talked to you about the function of transacetylase. Now, we are going to be seeing some mutant strains, and you've seen some mutant strains when we've talked about the Neurospora, that bread mold in one of the previous lectures. You've also seen some bacterial auxotrophs in which there was an indication by a negative sign of what they are incapable of producing, where they have a mutation. If cells are, in this system, if cells are incapable of producing functional beta-gal or functional permease, they are indicated as LAC-Z negative or LAC-Y negative. Okay? This simply means that they lack the ability to produce a functional protein of beta-galactosidase or permease. Okay? So we know due to bacterial chromosome mapping that the genes are in this order, ZYA, that is illustrated here. And now we're going to dive into exactly how these cis-acting sites and trans-acting molecules function to regulate the expression of this LAC operon and the structural genes within it. So here's the system that I've described to you, okay, with a few additional elements. You see the promoter and the operator. There's a leader sequence that we haven't really talked about. It's not terribly critical at this point. And then our structural genes. All right. So the three new things here are lactose. Remember, this is that raw fuel that the bacterium is capable of breaking down and getting the glucose it needs. We've got RNA polymerase. Of course, if we are going to transcribe a gene, we're going to need RNA polymerase. Now, the other new molecule here is this repressor protein. And as I mentioned already, this repressor protein is the product of expression of the repressor gene. So as I said, that repressor gene can be way down the line, it can be right here, it doesn't really matter. As long as there are repressor proteins floating around in the cell, they are able to do their job. And if you look at this repressor protein, it very clearly has two spots on it, a little notch on the top and a groove in the bottom. And these are sites that the repressor protein is able to interact with different molecules. The one on top, as you can see, is a perfect fit for the little spike sticking off of the operator, okay? And so this is the operator binding site. The repressor protein also on the bottom has a binding site that is a perfect match for what's sticking off of lactose. So this repressor protein can interact with the operator and or lactose, okay? Now this repressor is allosteric, okay? And what I mean by allosteric is that the way it looks right now with these binding sites that could bind to either the operator or lactose, those sites are changeable. Their availability, their ability to bind to the operator or lactose can change, and this change is an allosteric change. And it happens when there's an interaction with one of the binding sites. Okay, and we will see how that works here shortly. So let's look at what happens if in this system we have let's consider number B here on the bottom, okay? Look first at the genotype. It's I plus, O plus, Z plus, Y plus, A plus, okay? So Z plus, Y plus, A plus, ZYA, those are the structural genes. All of them are fine, they are the wild type, okay? O plus is the operator, okay? So the operator is the wild type. I plus is the repressor gene. It's also the wild type. So this is the normal way in which the lac operon works, okay? So what we have here is this repressor protein floating around, and you can see that it has bound to the operator. That little notch in the top fits perfectly with that little triangle hanging off the bottom of the operator. The repressor protein goes there and will attach to the operator, okay? Now the RNA polymerase matches with regions of the promoter, okay, in this upstream regulatory region of the operon. And when RNA polymerase latches onto the DNA, it's ready to transcribe. And it may start moving down and preparing to transcribe, but when it hits the operator that's locked up with that repressor protein, it can go no further. This repressor protein is a physical roadblock on the DNA that prevents the RNA polymerase from sliding down and transcribing the structural genes, okay? So this is a system where everything is functioning as it should, a wild type for every locus, and there is no lactose present in the system. Now think about why this makes sense. Do we want to make, as a bacterium, you are a bacterium, do you want to make beta-gal and permease and transacetylase? Do you want to spend a lot of energy making the stuff that allows you to break down lactose and let lactose into the cell and clean up the mess afterwards? Do you want to make all of those proteins if there's no lactose in the system to do it with, to break down? That would clearly be a waste of cellular resources and energy, okay? And so this system makes perfect sense. That repressor protein prevents the wasting of all of this energy in the production of proteins that aren't needed in the system. Now, if we change that to a wild type system in which lactose is present, we have a very different scenario. Look what happens here. Lactose actually interacts with the repressor protein binding to that bottom site. And when it does that, it induces this allosteric change to the operator binding site in the repressor protein. No longer does the repressor protein have that V-shaped notch in the top of it. It is now incapable of binding with that dangling triangle from the operator, okay? Because that repressor protein isn't there acting as a roadblock on the operator, it does not prevent the polymerase that attached to the promoter region from transcribing those genes. So it moves on without impediment, moves down, creates a messenger RNA molecule that is then translated by the ribosomes, and you end up with these polypeptides that become beta-gal, transacetylase, permease, okay? So this lactose actually served to induce the expression of this operon, okay? And because this transcription occurred when the repressor molecule failed to bind to the operator, the regulation is said to be under negative control, okay? So the lac operon is an inducible system that is under negative control, okay? Now you'll remember I talked about negative control. That is, expression occurs until turned off by a regulator molecule, okay? And that regulator molecule is the repressor protein, okay? All right. So what does this model of the lac operon suggest? It suggests that repressor genes produce a diffusible product. And what do I mean by diffusible? I mean that the repressor gene codes for and produces a repressor protein. And that repressor protein floats all around the cell and can interact with operons or operator regions, more specifically, anywhere in the cell. Okay? And so this protein is floating all around. It is diffusible. The operator is involved in regulation, but it doesn't have any gene product. Okay? And it has to be right next to the structural genes. Look at the way in which the operator works, right? You prevent transcription in the absence of lactose if that repressor protein is stuck to the operator. And it does that because the operator is right between the promoter, where the RNA sits down, and where the structural genes are. Okay? Look at this. The operator is right there in between the promoter and the structural genes. And so when that repressor protein roadblock sits there on the operator, it prevents RNA polymerase from transcribing the structural genes. If it weren't right there in between the two, it couldn't serve that roadblock function. Okay, so now we're going to look at a couple of mutant forms or strains that have mutations to both the repressor gene and the operator region. Okay, so in this first example, we have a mutant repressor gene that is producing a mutated repressor protein. This mutant repressor protein is not able to bind to the operator. Okay, if there's no repressor protein on the operator, it's not acting as a roadblock. It's not preventing the RNA polymerase from transcribing. So despite the fact there is no lactose present in the system, as you will see, there's no way to shut it off. It's going to run constantly. It will have constitutive expression. Okay, it's no longer inducible. Okay, it's always going. Another mutation that affects the operator binding site for the repressor protein. Okay, so in example B here, we have a normal repressor gene. The repressor protein is normal and functional. There's no lactose in the system, and so that repressor protein is able to bind to the operator, but when it gets there, the operator binding site is not compatible with repressor protein, and so no repressor protein binds. There's no roadblock preventing transcription, and so transcription takes place, and we get, again, constitutive expression. Okay. So this is a better view of that. I'm sorry, I should have advanced so you could see the bottom of this, but you're able to see the inability of the repressor protein to bind to this operator because the operator is mutated. Okay. Now, here's another interesting mutation to the repressor gene. Okay. We saw a mutation to the repressor gene that affected the operator binding site, but this is a mutation to the repressor gene that affects the lactose binding site. And so you can see here that the repressor protein binds to the operator, no problem. But in this system, we have lactose present. And so that lactose should be binding to the repressor protein at that bottom binding site. It should then create an allosteric change in the repressor protein, preventing it from attaching to the operator. Since there's a mutation to the repressor gene that, when transcribed and translated, results in a repressor protein that does not have a lactose binding site, there's no way to get this damned repressor protein off the operator or prevent it from attaching there in the first place. And because of that, there will never be expression of these genes. The RNA polymerase will never make it past the promoter region because this roadblock is permanently in place. Lactose can never remove it. Okay? And so the experiments that were done to test these hypotheses of the model are really quite fascinating. Okay? So the repressor protein gene produces a diffusible product, that the operator is involved in regulation but doesn't actually produce any product, and that it has to be adjacent to the structural genes. They were able to test this using F' plasmids to produce a merazygote. Now that should still be fresh in your mind, but we've got a bacterium that is F', so it's got its bacterial chromosome, but it also has a F factor that has a a gene on it, one of the genes. Maybe it's the repressor protein, maybe it's the operator, maybe it's Z, Y, or A. Okay? So they created these F prime cells and ran some tests to see whether these cells produced beta-gal when lactose was present or absent. Okay? And this is the most boring way to present this data, but it's very concise, very clear, and if you look at it and think about it, it makes complete sense. Okay? So I'll just go over the first few, but I encourage you to study this table and make sure you understand why things work out the way they do. In the wild type, the first line, all three portions of the lac-operon and this system are functional wild type genes. When you have a functional wild type repressor gene, operator, and LAC-Z gene, when lactose is present, the cell makes beta-gal. When lactose is absent, no beta-gal is made. Now let's look at example A, where the repressor gene and the operator are functional, but the LAC-Z gene, the beta-gal gene, is mutated and not functional. Well, of course, if the LAC-Z gene is not functional, you're never going to get any beta-gal, whether the lactose is present or absent. If, on the other hand, the repressor gene is mutated in the next line, the third line, the operator works, beta-gal works, this mutation to the repressor protein prevents it from binding to the operator, and therefore you're going to get beta-gal when lactose is present, as you would expect, but you're also going to get it when lactose is absent because the repressor protein is not able to bind to the operator. Okay? Therefore, it can't prevent RNA polymerase from moving down the gene region and transcribing those structural genes. So I'm going to jump to the first line, the second line of B, in which we have a mutated operator on the operon. So we have I plus O superscript C, so a mutant operator, and a wild type LAC-Z gene, and then slash F prime O plus. What that means is on the F factor, there's a functional copy of the operator. So it's not over by the ZYA genes, it's over on the F factor. So is it okay to have a functional operator within the cell if it's not near the ZYA genes? Okay, and you can look at the results here. You've got a plus in both columns, lactose present, lactose absent. That's not the way it's supposed to be. Okay, and so you should understand why it works out that way. And where it gets really interesting is down in C, when you change what's on the F factor and look at how that is able to rescue the function of this operon. Okay, so I encourage you to review this and maybe even draw it out. Draw out the bacteria, draw their chromosomes with the lac-operon and the alleles that they have for each of these genes or gene regions, or regulatory regions in the case of the operon, and draw what's on the F factor and think to yourself and make sure you understand why you get the results you see in this table. Okay, now it probably won't surprise you to understand that this, what probably seems like a very complex system of regulation, even though it's a very simple bacterial mechanism, is not the complete story. There's another regulatory molecule that we're going to talk about, and this is catabolite activating protein. Okay, and this molecule exhibits or exerts positive control over the lac-operon. Okay, so this is something that helps to activate expression, but it's inhibited when glucose is present. Okay, so this is an interesting point to think about, because up to this point, you've thought about the lac-operon and its function, whether it's being expressed or not, is completely controlled by whether there is lactose available, right? That's all that should matter. We don't make these things if lactose is not available because we don't need to break it down, okay? But if lactose is available, then yeah, we go ahead and transcribe and translate and make the stuff so that we can eat a bunch of glucose, right? But what we hadn't thought about is, all right, that's just whether lactose is present or absent. But what if lactose is present, but we already have a ton of glucose? We don't need any more. Is it worth transcribing and translating those genes when the only reason to do that is to make glucose and we've already got plenty of it? Okay? So you see why there's complex regulation of these genes. Here's how the catabolite activating protein works. All right? When glucose is absent, all right, this catabolite activating protein, so CAP, interacts with cyclic AMP. Okay? When the two interact, they form this, I'm going to call it cap-camp complex, the two of them together. And that binds to a specific site in the promoter region. You'll notice in this image, we've zoomed in on the promoter region. It used to only be wide enough that we could see the spot where polymerase sits down, okay? The operator is still over to the right. Now that we've zoomed in, we can see that there's a spot for the polymerase to bind, but there's also a spot for this cap-camp complex to bind. This complex only forms and then binds in the promoter region when there's no glucose, okay? When there's no glucose, we sure as hell want all of those structural genes made, because if there's lactose present, we want to make these gene products so we can break it down into glucose. So there's no glucose, cap-camp forms, and what cap-camp does is it really attracts or brings in RNA polymerase to that polymerase binding site in the promoter. Okay. If you don't have cap-camp there, it's just going to be the random chance encounter of a molecule of RNA polymerase with the promoter that leads to some transcription. But if you've got cap-camp there, you are going to get enormous attraction of lots of RNA polymerase molecules, and you will get lots of transcription. Okay. Now, when glucose is present, as you'll see on the bottom of the screen, cap does not bind with CAMP, okay, because CAMP levels are low. So there's plenty of glucose, there's not much CAMP, so cap-camp complex does not form. Cap can't bind to that site on its own without CAMP, okay? When it doesn't, RNA polymerase is rarely going to sit down on this promoter region that it can bind to. And so forgetting what happens on the operator with the repressor protein, if you have glucose, even if there's no repressor protein bound to the operator, and that would be, of course, because there's lactose in the system, then if there's glucose present, you're not going to get much polymerization. You're not going to get much transcription because there is not enough cyclic AMP for cap to bind to that promoter region site, okay? So the perfect world for maximal expression is no glucose is present, okay? The glucose is absent, as you see up top, and lactose is present, so you don't have a repressor protein bound to the operator, okay? Those two things are the perfect world. Lots of lactose, hey, we can break that down into glucose. Do we already have lots of glucose? No, we need it. So that's the perfect world. Lots of lactose, no glucose. We're gonna get loads and loads of expression here, okay? So remember, you need to be able to tie these examples back into negative and positive control and whether or not these are inducible, constitutive, or repressible systems. So let's summarize the lac operon and the way in which it can be controlled. And so here I've got all of these possibilities showing the different sites where the repressor protein can bind or the polymerase will bind, and where the cap-camp complex will sit down, okay? So in this first top example, we've got low glucose and there is lactose. Hey, that's the perfect world, right? We don't have glucose and there's the stuff to make it. we're going to go ahead and transcribe these genes. We get strong expression because there's no repressor protein on the promoter site, or the operator, sorry, and CAP-CAMP is sitting down in the promoter. You get RNA polymerase coming in at high levels, and you get loads of transcription. If in the second example we have high glucose, we don't need to make anything. Lactose is unavailable. Well, it's a good thing we don't need to make anything, because we don't have anything to digest to make glucose. So the repressor protein is on the operator, and there's no CAP-CAMP on the promoter. If we have low glucose, that means we're hungry. And there's no lactose. Bummer is, looks like we're going to stay hungry for a while as bacteria. Low glucose means we get the CAP-CAMP, but there's a repressor protein sitting on the operator site because of the absence of lactose. Okay. Now, if we have high glucose, so we're not hungry at all, but we could make more glucose, it's a waste of time. Okay. There's no repressor protein sitting on the operator. There's also no CAP-CAMP sitting on the promoter site. And so what we are going to get is we will get some very low levels of gene expression. Some polymerases are going to randomly sit down in the promoter just by chance and go ahead and transcribe because there's no repressor protein in the way, but it's not going to be much. All right. So in this, we've covered the lac operon pretty carefully and in a fair bit of depth. And I'm sure all of you will appreciate the fact that we no longer teach that in BISC 160, but it's something that we've retained in genetics for sure to help you understand how genes can be regulated. And this was one of the first systems that was described, and it is, as we said, an inducible system. Now the tryptophan operon was the first repressible system described. Now remember, an inducible system is one whose production has to be stimulated or turned on. A repressible system is one in which that production is on unless it's turned off. Okay? And the TRIP operon, for your reference as we go through and talk about it, is under what we call negative control. So it's a repressible system under negative control. All right. So here we are looking at the TRIP operon. So you can see there are 5 structural genes, E, D, C, B, A. Okay. And there is a regulatory region that consists of a promoter and an operator, a leader sequence. Again, we don't talk about that a great deal. And there is this new region, an attenuator sequence. So we'll talk about that in more detail here shortly. Okay. There is a repressor gene, okay, that produces this diffusible product. You can see that there's a repressor protein that has in it a tryptophan binding site. Okay. This is an allosteric protein that can be modified when it interacts with other molecules. So let's just look at how this system works. Okay. The first example at the bottom here, example B, is when tryptophan is absent. So the repressor gene is transcribed and translated, and we end up with lots of this repressor protein, but the only thing that protein can bind to in its naive, new baby form is tryptophan. Well, this is the gene region, or the operon, responsible for making tryptophan. And so since there is no tryptophan, what do you think we want to do? We want to express these genes. We want to make tryptophan because we're out, okay? So since that repressor protein is not attached to tryptophan, it maintains this shape with just one little notch on the left, and it is unable to bind to the operator site, okay? So since the repressor protein all by itself can't bind to the operator, we get transcription. We get expression of these genes, and we get the production of tryptophan. So that's the example that's here at the top. But when tryptophan is present, look at how this one works. Tryptophan binds to the repressor protein, but it does the opposite to the repressor protein that we saw on the lac operon. When this substrate, this tryptophan molecule interacts with the protein, the repressor protein, it changes it so that it can bind to the operator. Remember, in the case of the lac system, when the lactose binds to the repressor protein, it makes it so it can no longer bind to the operator. This is the opposite. When we have tryptophan present, it changes the repressor protein and makes it so that it does repress. Okay? This repression takes the same form as what happened in the lac operon when the repressor protein is bound to the operator. It sits between where the polymerase sits down and the structural genes, and it prevents transcription. Okay? So no tryptophan, you're going to get lots of expression. When tryptophan is there, you're going to block transcription. Now we're going to talk about the function of the attenuator and its role in regulating transcription of the genes in the trip operon. Okay? The way the attenuator works and the process of attenuation works is that, remember, we're trying to regulate whether or not a certain product is made, that product being tryptophan. Well, if tryptophan is present, remember, we shut down transcription. We've got plenty of it. It causes the repressor protein to bind to the operator, and we don't get expression. We don't get more tryptophan. We already have some. But what if there is a small amount of tryptophan present in the system, and so it might not get to the repressor protein because there simply aren't enough molecules of tryptophan present, but there are plenty present for the function of the cell. Well, in that case, that's where the role of the attenuator or attenuation comes in, okay? Because the way it works is no repressor protein bound to the operator and shut down transcription, all right? But what does happen is, since the repressor protein didn't block transcription, the messenger RNA starts making that, the RNA polymerase starts making that messenger RNA. And as it does, there is a portion in the region of the attenuator that is influenced, or it influences the process of transcription when tryptophan is present. Okay, so this messenger RNA folds into a configuration called the terminator that actually terminates the process of transcription. When it folds back upon itself, it shuts down the RNA polymerase. Okay? When there is no tryptophan present in the system, this terminator structure does not form, and the RNA polymerase slides right past the attenuator and proceeds to transcribe the structural genes. Okay? Now this system is described in your text. Again, I think all of these are ones that you would benefit from watching additional videos. Definitely read the text. Do some Googling that might help you. Okay? But this largely concludes our discussion of the regulation of gene expression in bacteria. Okay? Now, I want to summarize with something that I think will help you. I kind of created this to help think about positive and negative control and whether genes are inducible or repressible. Okay? So I think about it this way. A gene is under negative control under the scenario to the left. So does a repressor protein bind to the operator to prevent transcription? Okay? Or does an activator protein bind to DNA to stimulate transcription? Okay? So this, you can see the repressor protein in the lac operon exerts negative control, but the cap-camp complex is positive control on the lac operon. Okay? And then under negative control, you can read these definitions and see whether the system is either inducible or repressible. Okay? So just remember that genes are either inducible or repressible, but they can be under both negative control and positive control. Okay? So that would be two different molecules like the repressor protein and the cap-camp complex. Okay? So one exerting negative, one exerting positive control. Again, it's a bit complicated. You're going to want to think about it a bit. You're going to want to roll through those lac-operon examples to make sure you really grasp this, okay? And finally, I want to prepare you for discussion of chapter 16 just by saying that we've covered prokaryotic or bacterial regulation of gene expression in this chapter. We're going to talk about the additional mechanisms that eukaryotes, such as yourself, the other ways that we are able to regulate the expression of genes, okay? So while we have genetic mechanisms very similar to what's going on in the operons of bacteria, we have additional layers of regulatory capability that can finely tune whether or not certain genes are expressed.